this video is going to introduce the difference between differential and structural stains, and then we'll focus on the Gram stain, which is a type of differential stain. So remember that most cells are naturally colorless, and so we need to stain them in order to view them and study them. So we've already talked about simple stains and negative stains, which give us very limited information. They tell us the morphology and arrangement, and the negative stain also tells us cell size. But that's it. We can't really identify an organism based off of a simple stain or a negative stain. We need additional stains to provide additional information to help us identify an unknown organism, for instance. So a differential stain is used to detect differences between organisms. So is it gram positive or gram negative? Does it have mycolic acid in its cell wall or not? Okay, so we're looking at subtle differences between, in this case, bacterial species to further identify what they are. Structural stains are looking for the presence or absence of a very specific cellular structure, like a capsule or an endospore. Okay, so some organisms have a capsule, some do not. Okay, some can form endospores, some cannot. Okay, so with the structural stains, you're looking to see if that organism has that structure. All right, now these stains also show morphology and arrangement, okay? But they, like I said, they do provide additional information on top of that. And so typically, if you get an unknown organism or a sample in general, people will normally just do the Gram stain first because you get so much more information on top of morphology and arrangement. Okay, so let's talk about the Gram stain. So this is a differential stain based on the bacterial cell wall. So remember that gram positives have a really thick peptidoglycan layer, whereas gram negatives have a thin peptidoglycan layer. So they have an additional outer membrane, and that outer membrane contains porins. So the differences in the cell walls are going to result in gram positive cells staining purple and gram negative cells staining red. And we get those two colors based on the two colored stains that we use in this process. So there are 4 steps to the Gram stain. After you make your smear, so remember, a smear is when you've added your cells to a slide, you've allowed them to air dry, and then you've heat fixed them. That's where the smear process ends. Now we're going into the staining process. So you first add crystal violet, which is a simple stain. It has a positive charge, so it's attracted to the negative charge of the cell. And then you add iodine, which is a mordant. And so it essentially, I envision it like linking arms with crystal violet. So it helps it form this really large structure. Then you add a decolorizer, and that's actually going to be the differential step. Okay, we're going to walk through this a lot more here in a minute. But the decolorizer is going to be what allows gram negatives to stain red, because so far everything's purple, right? All bacterial cells have an overall negative charge, but we need to be able to differentiate between gram positive and negative, so we can't have everything be purple. So the decolorizer is going to essentially wash the purple away from gram negatives. So then we add the secondary stain, which I'll typically call the counter stain, which is safranin, which is red. It also has a positive charge, so it will now stain those colorless gram negatives red. So at the end of the day, we have purple gram positive cells and red gram negative cells. Okay, so why do gram positive and gram negative cells stain differently from one another? Well, it all comes back to that peptidoglycan layer and the presence or absence of a second membrane. So real quick, let's do a quick sketch of what's going on here. So remember that the, sorry, I'm trying to write and think, and it's not working for me. Okay, so both gram-positives and gram-negatives have a plasma membrane, right? That phospholipid bilayer. Now our gram-positives have a really thick peptidoglycan layer. For some reason, this highlighter is no longer working. Uh-oh. Now my computer's not wanting to work. Okay, there we go. Sorry, y'all. So it's got that thick layer of peptidoglycan. I'm just going to draw that part in yellow right there. But remember that the gram-negatives have a very small layer of peptidoglycan. So it's only going to have this little bit around it. The pen is working better than the highlighter. So let me kind of finish this one, because it's really going to make my point here in a second. All right. So because our gram-negative organisms have such a thin layer of peptidoglycan, they have that second phospholipid bilayer, that outer membrane, okay? So what's going to happen is you first add crystal violet to your smear, and because it's positively charged, both cells are going to be attracted to that dye, and they're going to become purple, which is what you see right here. We're kind of following along right there. So right now they're both purple. What happens next, let me see if I can kind of add purple to this. Okay, so at this point, the purple is all kind of in here. Okay, and it's looking nice and purple under the microscope. Okay, it's all in here. It's attracted to the cell. All right, and so then you add iodine, okay, which forms a crystal violet iodine complex. So that's making this even bigger, okay, making it even larger, kind of trapping it in the cell. So again, all the cells are still purple at this point. The decolorizer is where things change, because the third step, the decolorizer step, is the differential step. Because what's going to happen here is when you add alcohol to a gram-positive cell, it dehydrates that thick peptidoglycan layer. So imagine when something dehydrates, it kind of goes, and it kind of shrivels a little bit. And so what happens is that peptidoglycan layer kind of shrivels a tiny bit, like it tightens up, and it traps that crystal violet inside the thick peptidoglycan layer. So the gram-positives are forever purple, because the decolorizer, the alcohol, dehydrated that thick peptidoglycan layer, which traps the crystal violet inside. Okay, but when you add the decolorizer to a gram-negative organism, alcohol actually causes the outer membrane to dissolve. Okay, that's why with COVID, because it's enveloped, it has a phospholipid bilayer around it. That's why they say to wash your hands with soap and water and use sanitizer, specifically sanitizer in this example, because it has alcohol in it, and that will cause the envelope to dissolve, thus destroying the virus. So same concept here. You add the alcohol because it dissolves the outer membrane, and what that does is it causes all of the crystal violet to be washed away, because that tiny little peptidoglycan layer, it's like 1 or 2 layers thick. There's nothing to really trap the crystal violet. So at this point, the crystal violet is washed away from gram-negative cells. Okay, so now if you were to look at them both under the microscope, they would look differently. Your gram-positives would still be purple, but your gram-negatives would be colorless. And that's bad, right? Because from a person that's doing this perspective, right, if you look under the microscope, can you see colorless cells? No. So you'd be like, oh, did I do this process right? Did I just wash all my cells away and there's nothing there? Or are they just gram-negative? Right? You don't know. And so you don't want to guess if you did the procedure correctly. So that's where the counterstain safranin comes into play. You add safranin last, and it's going to be attracted to all the types of cells, but you just can't see it over the purple of gram-positive. Okay, so gram-positive you'll view as purple under the microscope, and gram-negative will now be red because the safranin will have been attracted to the cells. Okay, so again, the big difference here is the decolorizer step. It has two different effects on these types of cells. For gram-positive, it dehydrates that thick peptidoglycan layer, trapping the purple crystal violet inside, so the cell appears purple, whereas with gram-negative, the alcohol washes the outer membrane away, which washes away the crystal violet iodine complex, so the cell is colorless. So when you add the safranin, all you'll see is red under the microscope. So of all the stains, this is probably the most tedious because, as you'll see on the next slide, there's specific time requirements that each stain or step should be done. And really, it's the decolorizer step that can cause things to go wonky. So if you leave your decolorizer on your smear too long, then it can cause gram-positives to appear reddish because you're just saturating these cells, and eventually, right, a cell can only take exposure, or a cell can only take so much exposure to alcohol before it does start to affect the cell wall negatively. And so it can affect the color or appearance of those gram-positive cells. But if you don't leave the decolorizer on long enough, then you're not going to dissolve all those outer membranes, and you're not going to wash away all the crystal violet. So your gram-negatives will appear purple when they shouldn't. Okay, so leaving that decolorizer on for the right amount of time is key. Of course, you want to make sure that your smear prep was done properly. You don't want to have too many cells, because if you have a ton of cells in one spot, then they're going to be stacked on top of each other. And so when you try to rinse them, like with decolorizer and add safranin, those bottom level of cells may not get rinsed and stained properly. And so you want to make sure you have this nice, even, like monolayer of cells. And then you want to use fresh culture, because gram-positives, some of them, can lose their ability to retain the crystal violet iodine complex if they're too old. Okay? All right, so let's walk through the procedure really quick. There won't be a lot of procedural steps on your lab exam. You will need to know what the 4 steps are, like the crystal violet, then iodine, decolorizer, and safranin. But this is just so you get an idea of what you'd be doing if you were in the lab. So you've made your smear, which ends at the heat fixing step. And then you're going to add crystal violet onto your slide, and you let it just sit there and soak for about a minute. Then you're going to tilt your slide at an angle, and we do that using a clothespin so that we don't get covered in stain. So you tilt your slide and you rinse it gently with distilled water. You always spray along the top of the slide so the water runs down gently over your cells. You don't want to blast your cells with water. That would be like spraying us with a fire hose. Once you've rinsed off the crystal violet, you add iodine and let it sit on your cells for a minute. Tilt your slide, rinse with water. Then you're going to do your decolorizer step. And for this one, you want to have your water bottle right next to you, because you're going to tilt your slide, you're going to tilt your slide at an angle, and you're going to run the decolorizer, again, squirting at the top of your slide so it just runs down over your cells. And you do this for about 10 seconds until the runoff is clear. And then you immediately rinse with diluted water, because again, you don't want to leave the decolorizer on too long, or else you'll get some potentially false or confusing results. Once you've rinsed off your decolorizer, you're going to add the saffron in, let it sit for 1 minute, and then you are going to blot it dry with bibulous paper, and then you're ready to observe it under a microscope. All right, y'all, that is the end of this video. Let me know if you have any questions.